This is a super quick overview of the changes in metabolic approach present within cancer cells. And the readings for this topic are taken from the literature, and the PDF files for the readings are in the module on Canvas. On this side, you can see a histological section of the normal glandular epithelial appearance in pancreas. And here you can see example of pancreatic cancer. So we have proliferation of atypical cells marked by increased metabolic rate and increased rate of replication. Cancer cells tend to exhibit what's called the Warburg effect. And the Warburg effect is marked by increased rate of glycolysis even when oxygen is present. And so I should clarify this for you, and I should say that we have an increased rate of glycolysis followed by lactic acid fermentation. And there's really less running of the TCA cycle, and there's less running of the electron transport chain. So the ATP needs of the cell, the cancer cell, are being met by glycolysis, lactic acid fermentation. Glycolysis, lactic acid fermentation. These pathways give us a lower overall yield of ATP, but because the pathways are running with such high flux, there's a lot of ATP being produced. So cancer cells, even when oxygen is present, will not be running TCA and ETC. Note also that there's a lot of tumors that develop in hypoxic areas, especially if we're looking at solid tumors in soft tissue. And especially, if this mass of cells develops far ahead of the formation of blood vessels to bring in blood, right, and oxygen to the tumor. So this is an image that you've seen before to look at different metabolic approaches to meeting the ATP needs of cells. Normal healthy cells tend to run aerobic respiration. So they're going to run glycolysis, TCA cycle, electron transport chain, and then we have ATP synthase that's going to give us tons and tons and tons of ATP. Cancer cells, on the other hand, even when oxygen is present, are going to run glycolysis fermentation, and specifically, we're running lactic acid fermentation, which relies on lactate dehydrogenase enzyme. Now, metabolically, there are increased enzyme activities associated with cancers, and these cancer markers can be used to screen for presence of neoplasms or to screen for reoccurrence of cancers. And so these can be accomplished with taking a regular blood sample. So you can look for lactate dehydrogenase activity to be increased, hexose phosphate isomerase activity to be increased, and aldolase, among other enzymatic activity markers for cancer. Quick overview of the nature of cancer cells. So cancer cells are mutant. They are aberrant, meaning that they are abnormal. Cancer, there's a multi-hit hypothesis to development of cancer, and that there needs to be multiple mutations throughout the genome of the cell in order to remove all of the checks and balances on cell replication. And because of this, there's a lot of changes in the genome, and they could be, as far as genome changes, they could be change in DNA sequence, or they can be epigenomic changes, which in the last few years, 5 to 10 years, the importance of epigenomic signals in the genome are being much more highly recognized for their importance. And so epigenomic changes, one example there would be methylation patterns. So some DNA bases are going to be methylated and others won't be. And that has implications for what genes are turned on or off, meaning what genes are being expressed or not. Cancer cells are hypervariable, and so mutation begets mutation. So once we have a little bit of mutation and we lose control of the cell cycle and we lose that check to make sure that all DNA has been replicated appropriately, and we take a pause to allow the DNA replication machinery to catch up and fix changes. Once we lose those normal checks and balances, we're going to have increased rate of mutation within cancer cells. And so there's just a lot of change occurring in these cancer cells as a population. Also, as a population, these cancer cells are heterogeneous. If you take one tumor, you may find that there are multiple different genomes present in those cells that make up that tumor. Why? Because we have hypermutation in the cancer cells, and the cells are mutating. Different genomes are going to cause expression of different proteins, and so the cells will all be behaving a bit differently. Overall, cancer cells exhibit the Warburg effect, and that means that cancer cells prefer to use glycolysis followed by lactic acid fermentation with lactate dehydrogenase, even though oxygen is present. And again, the center of solid tumors can become hypoxic anyway if you consider that the rate of cellular replication is much faster than the development of blood vessels to serve the core of that tumor. Here's something interesting. Hypoxia is associated with resistance to chemotherapy and radiation. And so hypoxia, or being in a hypoxic environment, like at the center of a solid tumor, is an advantage to cancer cells because cancer cells that are in a hypoxic environment seem to have a greater resistance to chemo and radiation, so that hypoxia is a survival advantage to these cancer cells. Hypoxia is going to, again, force an increased reliance on glycolysis and lactic acid fermentation. So, because cancer cells are more heavily reliant on just doing glycolysis, lactic acid fermentation to support their ATP needs, if we hit cancer cells with drugs that target the glycolytic enzymes, now those cancer cells are even more sensitive to those glycolysis targeting drugs. So there's two sides here to consider. On one hand, hypoxia is making the cancer cells more resistant to chemo and radiation so that they have a survival advantage. On the other hand, hypoxia forces the cancer cells to be more reliant on glycolysis and lactic acid fermentation. So you could look at that second part as kind of an opportunity, right? So this is an opportunity to treat cancer with drugs that target glycolysis. And in theory, these glycolysis targeting drugs should have a good therapeutic index or a selective toxicity to the cancer cells because cancer cells are much more reliant on glycolysis than healthy cells. And so the cancer cells should be more greatly affected by these glycolysis enzyme targeting drugs than the healthy cells. So there should be a limited amount of damage to healthy cells and healthy tissue. As a reminder, you can see the early stages here of glycolysis, and then we have the final stages of glycolysis here. This circled portion here is lactic acid fermentation. And over here, you can see the conversion of pyruvate to acetyl-CoA, which then enters into the TCA cycle. This path here is greatly reduced in cancer cells. And so our cancer cells are running glycolysis fermentation. And remember, the only biochemical point of fermentation is to regenerate NAD plus so that we could continue to run glycolysis. And here is our glyceraldehyde 3-phosphate dehydrogenase, which is also commonly abbreviated as GAPDH. Arsenate is a strong inhibitor of glyceraldehyde 3-phosphate dehydrogenase because we have that deprotonated cysteine that is important at the active site of glyceraldehyde 3-phosphate dehydrogenase, and that can be complexed by many different metals. Over here, please note, we have glucose 6-phosphate dehydrogenase, which is the enzyme that controls the flow of carbons into the pentose phosphate pathway. The pentose phosphate pathway is kind of important to cancer cells because cancer cells need a lot of this ribose 5-phosphate in order to support formation of nucleotides to replicate DNA to support this high rate of cell replication. Cancer cells also would benefit from an increased amount of NADPH, which ultimately will be used to support a reduced population of glutathione in order to scavenge oxygen radicals within the cancer cells. Many glycolytic enzymes are multitaskers and they have more than one job within cells. Indeed, we're finding out more and more that enzymes have other roles than what we traditionally have characterized for them. So if we look at hexokinase, glyceraldehyde 3-phosphate, dehydrogenase, and enolase, all of these enzymes can also serve as transcriptional regulators, which can turn genes on or off. So they can change gene expression. And when you change gene expression, you change the proteome of the cell, meaning what proteins are present in the cell. When you change the proteins present in the cell, you change the metabolic profile of the cell. Hexokinase and glyceraldehyde 3-phosphate dehydrogenase are important in apoptosis. And I want to add in here cytochrome C. Cytochrome C is important for shuttling electrons from complex 3 to complex 4 in the electron transport chain. And normally cytochrome C is sequestered within the intermembrane space of the mitochondria. But when cytochrome C is released into the cytoplasm, cytochrome C can drive apoptosis. And apoptosis, of course, is a programmed cell death. And of course, if we have cancer cells, we want them to die. And so if we could upregulate apoptosis in cancer cells, that would be a good thing. On the other hand, cancer cells don't want to die. So if cancer cell metabolism is set up in order to prevent apoptosis, that's a benefit to the cancer cells. And finally, we have phosphohexose isomerase, which is able to enhance motility of cells. And if you think about motility as a benefit to cancer cells, cells which are motile are going to be more able to invade into other tissues. And so that ability to invade is required for metastasis. Taking a quick look here at hexokinase. There are 4 isoforms of hexokinase enzymes, and they do all of the same thing, meaning that they tag glucose with a phosphate on carbon 6 to trap glucose in the cell. Hexokinase 1, 2, and 3 all have a low Km for glucose, and so they have first dibs on glucose, as opposed to hexokinase 4, aka glucokinase in liver, which has a high Km for glucose and last choice, right, at glucose. So this is in liver, and this is other cells of the body. Hexokinase 2 specifically is increased in cancer cells. And the expression of hexokinase is increased because there's increased transcription due to promoter changes. And you could look at this and say mutation in the promoter of the gene. And the promoter of the gene is where we have RNA polymerase entering onto the DNA in order to start transcription of the gene. There are changes in the promoter sequence which cause RNA polymerase to bind more frequently and increase the transcription or the number of mRNA transcripts for hexokinase in the cell. So we get more mRNA transcripts, we're going to end up with more translation and expression at the protein level. Normally, hexokinase 2 is going to dock onto the mitochondrial outer membrane. And so it's going to dock onto, again, the mitochondrial outer membrane, so here, and the active portion of the enzyme is still in the cytoplasm, but when hexokinase 2 is docked on the mitochondrial outer membrane, apoptosis is inhibited. This is a benefit to cancer cells because cancer cells don't want to die. Note that hexokinase 2 docking onto the outer membrane is promoted by insulin. So could you say that insulin as a hormone is carcinogenic? Because insulin is going to promote the greater docking of hexokinase 2, the mitochondrial outer membrane, and inhibit apoptosis and also drive glycolysis, which is important for cancer cell metabolism. So cancer cells really like their glucose. And I recall reading some things, I don't remember exactly where, but reading some things about sugar being very bad for you because sugar can promote cancer, or high sugar consumption is associated with cancer development. Kind of makes you think. Here's an image from one of the papers that's on Canvas if you would like to read it. You can see here that when we have hexokinase bound to the mito outer membrane, we're going to have viable cells, meaning no apoptosis. Apoptosis is inhibited. And so when we have hexokinase in the cytoplasm, that means that apoptosis can occur. This is a situation in cancer cells. And so this is a benefit to the cancer cells. If we could somehow target hexokinase 2 and prevent its docking to the mitochondrial outer membrane, that would be a benefit to the treatment of cancer because, right, we want cancer cells to die. Also note here that cytochrome C present in the cytoplasm will induce apoptosis. Phosphohexose isomerase, which is important for the interconversion of glucose 6-phosphate and fructose 6-phosphate, is associated with cancer. Phosphohexose isomerase can serve as a metabolic marker for cancer. So if there's increased phosphohexose isomerase activity detected, it could indicate the presence of cancer. Phosphohexose isomerase, when released extracellularly, is able to promote motility and advancement of cells into other tissues, and that's necessary for metastasis. Glyceraldehyde 3-phosphate dehydrogenase does a little bit of everything. What doesn't this enzyme do? It's constitutively expressed, meaning that it is always transcribed and translated. It can act as a regulator of transcription and a regulator of translation because it has the ability to bind to DNA and affect gene expression, has ability to bind to RNA to affect the translation of mRNAs. It's important for endocytosis and membrane fusion. So it's important for taking materials into the cell. It's important for DNA replication and repair. It affects histone expression. And remember, the histone proteins are important as core proteins for wrapping DNA around those octameric histones in order to protect DNA and to organize it into chromatin. Glyceraldehyde 3-phosphate dehydrogenase, when it's present in the nucleus of the cell, is going to promote apoptosis. And so, glyceraldehyde 3-phosphate dehydrogenase is active in glycolysis in the cytoplasm.